All right, it's 12 o'clock and people are still trickling in. So welcome everyone to today's Connect Science seminar. And today it's my distinct pleasure to introduce to you Roberto Sonco, who's joining us from UC Berkeley. Roberto's lab really focuses on the fundamental questions around how nutrients shape and regulate growth and homeostasis. And he uses the lysosome as a model system to answer central questions around these. His lab has worked out many fundamental mechanisms of how nutrient sensing and the response to it happens, which all are of critical importance for our understanding of development, aging, and many diseases such as cancer and neurodegeneration. A few words on Roberto. He obtained his undergraduate degree from the University of Pisa in Italy before moving across the Atlantic to study neuro and cell biology at Yale, where he did his graduate studies with Pietro de Camilli. And after a couple of very productive years at Yale, he moved to Boston, so into our neighborhood, to the Whitehead Institute, where he worked with David Sabatini, and I think probably discovered his fascination for the lysosome and nutrient sensing, although very important for neurobiology as well. In 2014, he moved to Berkeley to start his independent lab. And at Berkeley, he is now an associate professor of biochemistry, biophysics, and structural biology in the Department of Molecular and Cell Biology. Across his graduate and postdoctoral studies, and since starting his independent lab, he had many seminal discoveries and consequently received prestigious prizes and awards, such as the NIH New Innovator Award, a Pew Scholar Award, and a Damon Runyon Radcliffe Innovator Award, and several others. And also, as I just learned about 2 weeks ago when I visited Berkeley, he's an expert on wine. So should you ever go out for dinner with him, let him choose. You can't go wrong. Roberto, I don't want to take much more time away from you. Just one final housekeeping item for all in the audience. If you have questions, please post them using the Q&A tool, and we'll read them and get to those at the end. All right, I think that's all my housekeeping. Roberto, the floor is yours.
Okay, thank you very much, Eric, for the very nice introduction and for the invitation. I'm really thrilled to present some of our work to the Dana Farber audience. So I will be talking about nutrient sensing and the role of the lysosome in this process. And these are my disclosures. So nutrient sensing, a major topic of interest in my lab, is the process through which the basic building blocks of life, such as amino acids, lipids, sugars, and nucleotides, not only contribute to building cell mass, but they can also give rise to signals that instruct the behavior of cells, organs, and organisms. For example, in response to certain nutrient cues, cells can decide to differentiate, or proliferate, or die. Organs can adjust their size in response to changing metabolic states of the body. And ultimately, nutrient signals can decide the trajectory of our growth and health, and eventually when we die. But nutrient sensing is a complex process because any cell in the body, for example, this neuron here, is constantly bombarded by very different chemical species, not only nutrients themselves, but also oxygen, energy, stressors, growth factors, and so on. So how do cells make sense of this chemical noise? So it's a complicated process, and it turns out that to solve it, eukaryotic cells have evolved sophisticated nutrient sensors that can integrate a wide variety of diverse stimuli and then generate precise instructions whether to grow or repair or proliferate and so on. So our favorite nutrient sensor is this large and ancient kinase called the mechanistic target rapamycin complex 1, or in short, mTORC1, which is conserved all the way from yeast to humans, and which really works as a central regulator of metabolism. In particular, mTORC1 promotes mass accumulation, so the process of growth that at a cellular level is a prerequisite for cell division. Cells have to double their size before they divide. mTORC1 is one of the major drivers of this process. And mTORC1 promotes growth or anabolism in response to a wide variety of upstream stimuli, which includes nutrients, as I mentioned, but also several growth factors energy, all of which are positive regulators of mTORC1. Whereas stressors such as DNA damage or starvation and so on can actually block mTORC1 and therefore prevent the cell from entering into a growth state when conditions are not favorable. And so the growth responses that depend on mTORC1 are the things such as protein synthesis, lipid synthesis, energy production. Whereas at the same time, mTORC1 is a strong suppressor of processes that consume cell mass, such as autophagy. Not surprisingly, given the central role of mTORC1 in cell metabolism, its dysregulated activity is implicated in many human diseases. Typically, too much mTORC1 activity is a driver of several cancers, can lead to type 2 diabetes, is a driver of aging, and can contribute to neurodegenerative conditions. And so one of the major goals in the field is to really understand mechanistically how mTORC1 is regulated so that we can come up with better strategies to control or rein in its activity. So probably the most remarkable discovery in the past 10 to 15 years has been the identification of the cellular site where mTORC1 becomes activated. This has now become almost a paradigm where nutrients, particularly amino acids, as these initial discoveries were made, cause mTORC1 to relocalize from the cytoplasm, where mTORC1 is inactive in low amino acid states, to the surface of an organelle, the lysosome, when amino acid levels in the cell are high. So we get this amino acid-driven relocalization and association of mTORC1 to the lysosome. We can take any cell type of choice, for example, HeA cells shown here, start them for amino acids and stem for mTOR, which is diffused and doesn't colocalize with this green LAMP2 lysosomal marker. We can then stimulate with amino acids, and within a few minutes, you can see how mTORC1 goes from diffuse to this beautiful cluster localization, which corresponds exactly to LAMP2 positive lysosomes. In fact, mTORC1 doesn't seem to go anywhere else. Really, the lysosome seems to be the site of its activation. And so many studies have essentially supported a model of signal integration, where nutrients such as amino acids, but also others, as we will see, position mTORC1 on the limiting membrane of the lysosome. And there, the kinase function of mTORC1 can be unlocked by other orthogonal signals brought by growth factors, for example, insulin, PI3K, AKT signaling, so that now mTORC1 can phosphorylate a number of substrates that are involved either in upregulation of anabolic processes or downregulation of catabolic processes. And so this model ensures that mTORC1 drives the cell into a growth trajectory when both local conditions signaled by nutrients and long-range signals brought by growth factors are available. There are interesting features about this model. Not only told us about mTORC1 itself and how it's regulated, but it's actually taught us a lot about the lysosome. So first of all, the lysosome is not just a backdrop where mTORC1 becomes active, it actually participates in conveying nutrient stimuli to mTORC1. And this is the case for both amino acids, cholesterol, as I will discuss, and probably other nutrients as well. Moreover, upon its activation, mTORC1 can exert feedback regulation on the lysosome itself. For example, mTORC1 is a strong regulator of a transition factor called TFEB, which I will mention later in my talk, which in turn functions as a master regulator of lysosomal biogenesis, where it can promote the proliferation of lysosomes, increasing their catabolic potential and therefore cell catabolism. So mTORC1 suppresses TFEB, preventing therefore lysosomes from exerting too much catabolism in the cell. So given these activities, we now really have changed our views of the lysosome from the recycled bin that's commonly described in all textbooks to a sophisticated signaling center that can sense and integrate metabolic cues, relay them to mTORC1 to then essentially drive the cell toward a trajectory of growth or repair. So given this, there are major questions that remain open and that my lab is actively interested in investigating. One is which nutrients activate mTORC1 and how. So I told you that amino acids are potent regulators of mTORC1, but we know that sugars, lipids, nucleotides also regulate this kinase. And so these are very diverse chemical species. How are they integrated to provide the proper level of mTORC1 activity? The second is, what are the mechanics of mTORC1 recruitment to the lysosome? How does it access the substrates? Are they all phosphorylated at once, or are there criteria as to which programs are going to be activated? So this is an area where we really don't know everything. And third, can we develop, based on this detailed knowledge of how mTORC1 is regulated, new strategies to control its activity, either wholesale or towards specific substrates in the context of specific diseases? And so I will touch upon each one of these three aspects during my talk. So first of all, when I started my lab, one problem that we became very interested in was how lipids regulate mTORC1. And the reason for this question was that it was known for a while that if you take mice and you put them on a high-fat diet or a high cholesterol diet, and you measure mTORC1 activity in peripheral tissues, you can see a clear upregulation of mTORC1 activity in response to lipids. However, it wasn't clear whether this upregulation corresponded to a cell-autonomous mechanism for lipid sensing. So how does mTORC1 actually sense lipids? And so we focus on cholesterol because we know the cells really care about cholesterol. This is a building block for membranes. It's a precursor for steroid hormones. And the Laszlo, Brown, and Goldstein showed beautifully how cholesterol can be sensed homeostatically at the ER by a dedicated sensing mechanism, which includes the SCAP sensor and the SRBP transition factor. So that cells can sense when cholesterol are low, and they can homeostatically increase its rate of synthesis to keep it within an optimal range. So we think of that as a homeostatic feedback mechanism, but mTORC1 is a master regulator of growth, where more membranes need to be made, the organelles need to be replicated or copied. And so could it be that cholesterol can also drive growth? And can it do so cell-autonomously via mTORC1? So the key findings that we made was that cholesterol can indeed strongly activate mTORC1. And in particular, just like amino acids, it promotes the localization of mTORC1 to the lysosome. So if we take cells and we deplete them for cholesterol and we stain for mTOR and LAMP2, so the marker for lysosomes, we can see that in low cholesterol states, mTOR is diffused and not found on lysosomes. However, as we supply cholesterol back to cells, we can see again this beautiful clustering of mTORC1, which again localizes or concentrates on the lysosomal surface. And this wasn't a given because many other stimuli that regulate mTORC1, for example, growth factors or energy stimuli, don't control its localization, but rather its kinase activity. And so this data place cholesterol in the same class as amino acids, so nutrient inputs that regulate the lysosomal translocation. So how does this happen? You know, when we think about lipids, such as cholesterol, they can exert many different forms of action, some of which are actually biophysical. For example, cholesterol could control membrane fluidity or some other property that favors mTORC1 recruitment. But actually, that was not the case. So our data indicate that this is an exquisite and very specific mechanism for sensing cholesterol itself and not some properties of the membrane. And so some evidence that that's the case comes from this experiment here, where we start the cells for cholesterol, and then we look at mTORC1 activation by either cholesterol itself or chemical analogs of this molecule. So here, when we deplete cholesterol, we had a shutdown of mTOR signaling, shown by loss of phosphorylation of this canonical substrate, SSK1 and 4-EDP. We can then reactivate mTORC1 by supplying the cell with either cholesterol itself or LDL particles. These are cholesterol carriers, and you can see this nice reactivation of mTORC1. However, if we now supply chemical derivatives that differ from cholesterol because of stereochemistry or addition of one hydroxyl or methyl group, now you can see how mTORC1 reactivation essentially fails. So mTORC1 is really attuned to sensing cholesterol and not other sterol-like molecules. Moreover, we were able to involve or implicate specific proteins in sensing of cholesterol. And so a key component here are these dimeric GTPases known as the RAG GTPases, which are in fact already obligate regulators in the amino acid sensing pathway. So here you have RAG-A in complex with RAG-C, and I will tell you more about this later in the talk. And like other GTPases, the nucleotide state of these proteins is key. So in particular, when RAG-A is GTP loaded, that's the activating state toward mTORC1. And so in similar experiments to say manipulations of KRAS, we can make mutants in RAG-A that are locked with GTP. And so these are constitutively activating toward mTORC1. We can then ask whether by making a constitutively active mutant of RAG-A, we can bypass the cholesterol starvation that I showed you before. And so here are the data. So in cells expressing the wild type RAG-A, we see that mTOR is diffused in the absence of cholesterol, and it clusters when cholesterol is added back to cells. However, in the cells expressing the GTP-locked RAG-A mutant, you can see how mTOR is already clustered on lysosomes even when cholesterol is taken out, and not much changes when we add it back. And this is quantified here, where you can see that the mutant essentially cancels out the differences between starved and refed. So we think that this is a remarkable result because you can trick the cell, or at least trick mTORC1, into thinking that cholesterol is there when it's actually not, just by making a single point mutant in the G domain of the RAG-A GTPase. So again, this speaks to a specific dedicated signaling pathway. So in fact, we discover through both functional experiments in cells and reconstitution in vitro, we discovered many details and many aspects about this cascade. For example, we know that the concentration of cholesterol on the limiting membrane of the lysosome is key. The lysosome actually has other internal membranes, but that cholesterol doesn't seem to matter. So it's really the cholesterol on the limiting membrane that matters. This cholesterol affects the nucleotide state of the RAG GTPases through various mechanisms, leading to mTORC1 recruitment. And an important aspect is that there are proteins that span the lysosomal membrane and therefore can come into direct contact with cholesterol so that their conformation can be modulated and translated into regulation of the RAGs and in turn recruitment of mTORC1. One protein we implicated a few years back is SLC389. This is an amino acid permease and a sensor for arginine in the mTORC1 pathway. But we discovered that SLC389 also harbors two canonical cholesterol binding motifs in one of its transmembrane helices, and these motifs are key for the ability of cholesterol to activate mTORC1. More recently, we discovered a new protein in the lysosome, which we call LYCOS, which stands for lysosomal cholesterol sensing. This is work in progress by Regina Shin, a fantastic postdoc in the lab, where again, within the cholesterol can modulate the conformation, so the three-dimensional structure of lysosomal proteins, resulting then in activation of the RAG GTPases. And so again, this is very much work in progress that we are very interested in understanding into great detail. Another question that we've been asking is, where does this cholesterol come from? So where is the cholesterol that activates mTORC1 generated, or how is it provided? So we know the cholesterol can reach the lysosomal membrane from the extracellular space when LDL particles are taken up and internalized and then disassembled. But a recent theme is that the lysosome is also in physical communication with other organelles, such as the ER, the Golgi, and mitochondria, through physical junctions through which lipids, which are insoluble and hydrophobic, can be exchanged via dedicated machinery. And in fact, the contact between the lysosome and the ER is actually quite prominent and quite fascinating. So here I'm showing you a movie of red lysosomes together with the two markers for the ER in green and blue. And you can see how really intimate the connection is between these two organelles. Literally, the lysosomes are wrapped in ER membranes, and they had to make their way through the ER network as they traffic through the cell. So there are several really cool examples of this intimate association. And so a former postdoc in the lab, Chunyan Lim, who is now an independent investigator in Guangzhou, China, discovered, in fact, that physical junctions between the ER and the lysosome are essential for the ability of cholesterol to activate mTORC1. In particular, Chunyan identified a tag team of proteins, one called VAP, which sits on the ER membrane, such as a transmembrane protein, and the other one called oxysterol binding protein, OSBP, which bridge the space between the ER and the lysosomal membrane, and they transfer cholesterol unidirectionally from the ER to the lysosomal membrane. So the main cholesterol transporting protein in this team is OSBP, which has a C-terminal folded domain that can essentially accommodate cholesterol and shield it from the hydrophobic environment of the cytosol and then deposit it on the receiving membrane. This is done in exchange for a phospholipid, the I4P, so it's a energy favorable process. Chunyan was able to show that there is a pool of the OSBP protein in green that associates with LAMP2 positive lysosomes in red, so you can see here the merge. And we find indeed that the transfer activity of this complex is really essential for mTORC1 activation. So this is shown with one of those experiments that we've seen now so far. So this recruitment of mTORC1 to the lysosome. So again, in control cells, where we knock down luciferase, in the cholesterol starved state, mTOR is diffused, not found on these LAMP2 positive lysosomes. As we add cholesterol, we see clustering of mTORC1 at the lysosomal surface. However, in cells where we knock down OSBP, we see a complete failure of this process. In fact, mTORC1 completely fails to go to lysosomes, and this is actually one of the strongest manipulations that inhibit mTORC1 localization that we could find. And it really contrasts with the mutant I showed you before, where we can make mTORC1 recruitment constitutive. This is also very specific to OSBP. So OSBP is only one member of a much larger family of proteins called the ORPs, whose job is to transfer cholesterol and phospholipids across organelle contacts, but only OSBP was specifically required for the lysosomal recruitment of mTORC1. And so this speaks to the fact that this is, again, a dedicated process that requires cholesterol transfer across the ER lysosomal membrane. In fact, we, again, through many experiments that I'm not going to, we discovered, again, many interesting details about this process. So first of all, we found that the cholesterol transfer activity of ApoSBP contributes a significant fraction of total lysosomal cholesterol, about 30%. So some of the cholesterol has to be continuously supplied from the ER. We found that the RAG GTPases are indeed involved. So I won't show the data, but we can rescue the loss of OSBP by making, again, those point mutants in RAG-A. And so we can make mTORC1 insensitive to this pathway. And so this told us essentially that this is, again, a dedicated pathway that converges on the RAG GTPases. We also think that this could be just the tip of the iceberg. So the fact that the lysosome engages in contacts with different organelle species and the fact that mTORC1 plays such an important role in cell metabolism suggests that perhaps this is one way in which mTORC1 can sense the status of other organelles. They are in this particular case, but perhaps also the mitochondria, the Golgi, and so on. So through the transfer of specific lipid species that can then talk to mTORC1 and modulate its activity and its downstream responses. We know that cholesterol regulates mTORC1, but there are hints that other lipid species might as well. And so there are many more aspects that we are excited to investigate in this process. So back to the model I showed you before, where this tag team of VAP and OSBP transfers cholesterol to the lysosome to enable mTORC1 activation. Of course, for every pathway that has an activating input, there has to be also an inhibitory input. And in this particular case, how is cholesterol removed from the lysosome to modulate mTOR activity? So it turns out that lysosomes harbor a well-characterized, or well-known at least, cholesterol transporter, the Niemann-Pick C1 protein, or NPC1. So the job of this protein is to mobilize cholesterol from the lysosomal internal membranes and limiting membrane, and then transfer it to acceptor organelles or membranes, including the ER, the Golgi, and the plasma membrane. And the activity of NPC1 is really essential. So we don't understand exactly how NPC1 actually moves the cholesterol off the lysosomal surface, but thanks to recent structural studies, we understand at least how cholesterol is loaded into the transmembrane domains of NPC1 to be further moved out. And the importance of this protein is highlighted by the fact that mutations that cause the loss of the functional NPC1 protein cause a devastating disease known as Niemann-Pick type C, which is characterized by extensive neurodegeneration, metabolic imbalance, and early death. We know that cholesterol levels accumulate massively in these NPC1 negative lysosomes, but we don't completely understand how this initial accumulation of cholesterol then results into toxic cellular processes or disruption of cellular homeostasis that then leads to all the various phenotypes or manifestations of this disease. So given what I told you, that cholesterol is a potent activator of mTORC1, a natural question that we asked was whether in cells that lack NPC1, whether there will be a change in mTORC1 activity. And actually that turns out to be the case. So on the left-hand side here, we have wild-type cells. We did these experiments in 293Ts, but also in many other cell types where the results are very consistent. So again, like I showed before, we can switch off mTORC1 by removing cholesterol. We do this by various means. And we can then reactivate it through cholesterol stimulation or LDL. In cells that lack the NPC1 protein, now mTORC1 is constitutively on, so we cannot turn it off by removing cholesterol. It seems that due to the high concentration of cholesterol in the lysosome, mTORC1 is always locked in the on state. We can then re-express NPC1 in these cells, and we can resume the normal mTORC1 regulation by cholesterol levels. In fact, the more NPC1 we express, the less activity of mTORC1 we can see, suggesting again a role of NPC1 as a negative regulator of mTORC1 signaling. So what are the consequences, though, for NPC pathogenesis? So given what I told you that mTORC1 has a huge influence on downstream cellular processes, could it be that hyperactive mTORC1 caused by the massive accumulation of cholesterol in these lysosomes could drive some of the cellular phenotypes? And so to identify or to be able to answer this question, we first had to take stock of what is wrong, essentially, in NPC1 defective cells, and particularly lysosomes, but also other organelle systems. So for this, we applied a technique that our lab and other labs had developed, and this was worked by Oliver Davis, a recent and terrific grad student from the lab. And so Oliver expressed a affinity tag in lysosomes, which we can then pull down, capture on the surface of beads, and then use either proteomic or lipidomic analysis to get a complete breakdown, essentially, of the proteins and metabolites that are present in these organelles, and also what differences are there between the wild type and the NPC1 defective lysosomes. So this technique works very well. So we can capture with high efficiency the majority of the lysosomes in the prep, so found here through this LAMP1 marker in the IP lane, whereas contaminating organelles such as mitochondria, ER, early endosomes are maybe left behind. And these lysosomes are intact. So if we preload cells with the fluid phase marker, dextran, Oregon green, that accumulates in the lysosome, the marker is still present after purification, indicating that these lysosomes did not break or pop or explode. So we can literally figure out not only what is present on the limiting membranes, but also what's present in the lumen. So the experiment we did was to compare lysosomal proteomic composition from wild-type cells, as well as cells with deleted NPC1, and we used two conditions. One is DMSO, the other one is treatment with protease inhibitors that cause the proteins being digested in the lysosome to stabilize. So by looking at the selective stabilization of proteins, we can figure out essentially what is the cargo being digested and the efficiency of this process. So the resulting data look like volcano plots here, where on the right-hand side is the enrichment in the protease inhibited condition, and all these red dots are the ones that significantly accumulate. And so in this population, you can find several of the proteins that are known to be degraded by the lysosome, and this includes many autophagic adapters such as sequestosome, GABA-RAP, and so on. So we then did this comparison where we look at the relative stabilization in the wild type versus NPC1 defective lysosomes, and we found something quite striking, which is that all the bona fide lysosomal substrates were accumulating to a large extent in the NPC1 lysosomes compared to the wild type. And so this was a telltale sign of a proteolytic defect where proteins have been imported into the lysosome, but then they're just sitting there, they're not being degraded. In fact, we could directly verify that by immunofluorescence. So here I'm showing you high mag details of lysosomes from wild-type cells here on the left, as well as cells lacking NPC1. And we co-stain these cells with LAMP2 in green and with a marker for autophagy, LC3B in red. So this is a typical canonical substrate that's degraded in lysosomes. So in the control cells or wild-type cells, there is not much LC3 accumulation in lysosomes. Whereas on the contrary, cells lacking NPC1 are chock full of undigested LC3. So you can see literally how LC3 is deposited and sequestered in the lumen. Now we can reverse this phenotype by re-expressing NPC1 in these cells. As you can see here, now the accumulation of LC3 is clear. However, if we add back into these knockout cells mutants of NPC1 that are incompetent for cholesterol transport, you can see how, again, we have this massive accumulation of LC3 in the lysosomal lumen, indicating that the cholesterol export activity of NPC1 is key. And this is quantified here. So in fact, we extended this proteomic analysis to all the aspects that we could find of lysosomal function and integrity. And we discovered through proteomics that not only proteolysis was defective, but also several hydrolases involved in digesting lysosomal cargo macromolecules are actually decreased in NPC lysosomes, and that includes not only proteases, but also lipases, nucleotidases, and so on. Moreover, these lysosomes are more fragile, so they actually break more easily when they're subject to chemical or mechanical stress. So these are all, again, telltales of profound lysosomal dysfunction that is a consequence of the cholesterol buildup that occurs. So we then asked whether this hyperactive mTORC1 that's caused by cholesterol accumulation would have anything to do with these phenotypes. And that was indeed the case. So here we're looking back at the accumulation of LC3 in lysosomes, which is the telltale of defective proteolysis. And so again, in the wild-type cells, we don't see accumulation of LC3 inside lysosomes. In cells lacking NPC1, these defective lysosomes, which are also large, are clearly full of undigested LC3. And here we inhibited mTORC1 via two different inhibitors, taurine-1, which is an ATP competitive inhibitor, and rapamycin, the famous allostatic mTORC1 inhibitor. So taurine-1 was quite striking. It completely cleared out the intralysosomal accumulation of undigested LC3. On the contrary, rapamycin completely failed to do so. You can see here how these lysosomes are still full of undigested LC3, and this is quantified here. So this told us two things. One is that mTORC1 inhibition can restore proteolysis, can reverse at least some of the defects of lysosomal function caused by loss of MPC1, but also that the way you inhibit mTORC1 really matters. And I will go back to this at the end of my talk. So in fact, to summarize a lot of work we did, we found that several aspects of lysosomal dysfunction that I described in cells lacking MPC1 were corrected by chemical or genetic mTORC1 inhibition. So by blocking mTORC1 in MPC1 null cells, we were able to restore proteolysis, so protein degradation. We were able to boost the levels of hydrolases, and we also restored the integrity of these membranes. So these lysosomes were no longer prone to permeabilization, which, by the way, can be itself very harmful to cells because all these hydrolases will be released in the cytoplasm. What mTORC1 inhibition did not correct, however, was the accumulation of cholesterol and other lipids in these lysosomes. So these lysosomes are still full of cholesterol. However, we can decouple this initial accumulation of this lipid from the downstream consequences in terms of proteolysis and lysosomal functionality. In fact, the benefits of mTORC1 inhibition went past that. So another typical sign of MPC1 defective cells, and this is actually common to many degenerative diseases where the lysosomal function is compromised, is the accumulation of damaged mitochondria. So the idea is that when the lysosome is defective, because of failed proteolysis or failed acidification, damaged mitochondria cannot be efficiently cleared by mitophagy and accumulate in the cytoplasm, thereby causing further damage that can be especially harmful to neuronal cells. And so this is indeed the case for MPC1. In fact, by using mitochondrial assays and also proteomics, we were able to demonstrate that the health of mitochondria is profoundly compromised. However, inhibition of mTORC1 via various means was sufficient to restore healthy mitochondria as shown by their morphology, membrane potential, and so on in cells lacking MPC1. And we were able to show this in a variety of cell lines, and particularly important in iPSC-derived neuronal cells. So then this leads us to a model that might have important therapeutic implications. So as I mentioned before, mTORC1 has major functions in several aspects of cellular behavior, including growth and proliferation, energetics, quality control, and so on. In processes where the lysosomal function is compromised, such as in Niemann-Pick type C disease, the initial accumulation of a nutrient that stimulates mTORC1 can lead to aberrant activity of the kinase and therefore disruption of these downstream processes, leading them to cellular and organismal pathogenesis. In Niemann-Pick type C, the current gold standard for therapy is actually active removal of cholesterol, which is performed by essentially injecting patients with cholesterol removing agents in the cerebrospinal fluid. So this is actually a very invasive procedure that is beneficial, it's working, but it's probably not, perhaps not sustainable in the long term. And so one exciting possibility from this data is that mTORC1 inhibition, while not sufficient to clear the cholesterol from these lysosomes, could uncouple this initial storage from several of the downstream harmful consequences, and so perhaps be sufficient to sustain cell homeostasis in various cell populations. So we're excited to explore this possibility with collaborators. So the second question that is further motivated by this data is how are mTORC1 recruitment to the lysosome and signaling regulated? So as I mentioned, we still don't understand many aspects of the mechanics to which mTORC1 is recruited, how its substrate is selected, and this could have profound implications for disease settings, such as the one I described. And so key to recruitment of mTORC1 are the RAG GTPases that I mentioned before. So I said how the RAGs are heterodimers, where RAG-A is in complex with RAG-C. And the RAGs are not directly anchored to the lysosome. They require a second complex, the pentameric regulator, one of which subunits is lipidated and essentially anchors the whole thing to the lysosome. And so key property of the RAGs is that they can switch the nucleotide state in a nutrient-dependent fashion, so that in low nutrients, RAG-A is GDP loaded and RAG-C is GTP loaded. And in this state, the RAGs cannot bind to mTORC1 physically, and therefore mTORC1 remains in the cytoplasm in the inactive form. On the contrary, when nutrient levels rise in the cell, the RAGs switch their nucleotide states through mechanisms that we'll talk about, so that RAG-A becomes GTP loaded, RAG-C is GTP loaded, and under these conditions, the RAGs can bind to mTORC1 and anchor it to the lysosomal membrane. So this is a cartoon view of the process. There is now beautiful structural work from the labs of from the Sabatini and Roger Williams labs that have shown the three-dimensional structures with atomic detail of how the RAGs actually anchor physically mTORC1 to the lysosome. So how is this switch affected? So how do the RAGs switch from this inactive state to the active state? So because there are 2 RAGs and 4 possible nucleotide states, we don't exactly understand how we get this transition from the inactive to the active state. There could be different routes through which this proceeds. Because these are GTPases and they are part of the RAS superfamily, they could perhaps be exchange factors that remove the GDP on either RAG and replace it with GTP, but this has not been conclusively identified. But we know for sure that there are GTPase activating proteins or GAPs, the job of which is to actually help convert the GTP to GDP by accelerating GTP hydrolysis. For example, GATOR1 is the main gap for RAG-A, which switches RAG-A back to the inactive state. So GATOR1 is required for the inactivation of mTORC1 when nutrient levels go down. And a second gap, which is composed of the folliculin FNIP complex, is involved in switching RAG-C from the GTP state to the GDP state, which is thought to be a step required for the switch of the RAGs to the active mTORC1 recruiting state for STEP. And so we focus on folliculin FNIP for a number of reasons. So as I mentioned, biochemically, this protein complex is a gap that promotes GTP hydrolysis and so release of phosphate on RAG-C. And it was proposed to be required for mTORC1 activation by nutrients. But there is a conundrum, which is that folliculin genetically acts as a tumor suppressor. In fact, its loss is associated with an inherited tumor syndrome, Verhoeven-Dubé, which is actually a precursor to renal cancer. And it actually resembles tuberous sclerosis in some aspects. So we get this dichotomy between a biochemically activating role and a genetically tumor suppressing role. So we wanted to understand how this could happen. So a further interesting aspect of the folliculin FNIP complex is its recruitment to the lysosome. So if we start cells for nutrients, amino acids in this case, we can see folliculin clustering on LAMP2 positive lysosomes. However, as cells are re-stimulated with amino acids, we see the folliculin becomes dispersed. It's no longer found on the lysosome. So this is actually the opposite behavior from mTORC1. mTORC1 is not on lysosomes in the manus amino acid states, and it clusters on lysosomes when amino acid levels go up. Folliculin seems to do quite the opposite. So we have this complex here on the lysosome that might have an important function. We wanted to know, what is it doing there? So what are these clusters, in other words? And so for that, we actually turned to biochemistry and structural biology. So we purified the main components of this complex, so folliculin and FNIP2. There are 2 paralogs, FNIP1 and 2. So we focus on FNIP2 for these experiments, together with the RAG-GTPases and the regulator complex. And so to gel filtration, we were able to show that these proteins associate in a very stable complex that can be isolated and purified by gel filtration. And an important aspect that I will touch upon in a minute is the fact that this complex only assembles when RAG-A is loaded with GDP, not with GTP. So that corresponds to the nucleotide, so to the inactive nutrient low state. In collaboration with Jim Hurley at UC Berkeley, we were able to then solve the structure of this large complex composed of folliculin, FNIP2, RAGs, and regulator via cryo-EM to about 2.4 angstrom resolution. And so this turns out to be a really beautiful complex where we can see that folliculin in green and folliculin in yellow or orange and FNIP2 in green are a tightly bound dimer that essentially is locked in the space between the G domains of RAG-A and RAG-C, whereas regulator projects on the other side. The lysosomal membrane will be down here. So we have a complex that perhaps projects upwards, where all these components are tightly bound to each other. As I mentioned, biochemically, folliculin FNIP2 is a gap. And so one natural question is whether this complex that we purified and solved represent the gap active complex. So in order to be able to determine that, we needed to know which one is the catalytic arginine in this complex. So just like every other gap for small GTPases, follicular FNIC2 we thought should have a so-called arginine finger, which is inserted in the G domain of the client GTPase and helps with the reaction, with the GTP hydrolysis reaction. And so through homology studies with then recently published structure of the GATOR1 complex, so the other gap, the gap for RAG-A, we were able to identify a putative catalytic arginine, which is in folliculin. It's present at the end of this loop here, projecting into the nucleotide pocket of the RAGs, R164. So first, we got very convincing functional evidence that this R164 is the catalytic arginine. In fact, if we do a gap assay in vitro, we can do it through various ways. Here we can see that the wild-type folliculin enables complete conversion of GTP to GDP in RAG-C. However, if we mutate the R164 to an alanine, we see complete failure of the gap reaction, indicating that most likely this is a catalytically essential arginine. So now, based on the functional data, we went back to the structure and we asked, where is this arginine 164 position in the complex? And so this is a close-up of the folliculin loop where arginine 164 sits. Here we have the RAG-C and the RAG-A nucleotide pocket. And we're actually quite struck by the observation that this R164 is actually located a full 23 angstroms away from the nucleotide in RAG-C, which is the natural substrate. And it's also about 27 angstroms away from the nucleotide in RAG-A. So in other words, in this complex that we isolated and solved, there is no way that this catalytic arginine could participate in catalysis. So this complex could not represent an active gap state. And in fact, through additional experiments, both in cells and in vitro, we came to a model where we think that this complex, which we call the lysosomal folliculin complex, or LFC, represents an inactive state of the complex, which is poised for activation by additional signals. And in particular, when nutrient levels are low, and so when RAG-A is loaded with GDP, this is sufficient to hold folliculin FNIC2 in a state that's tightly bound to the RAGs, but unable to perform the gap function. When nutrient levels go up, and so RAG-A becomes loaded with GTP, this is sufficient to break the complex, and it allows folliculin FNIC2 to reorient in a way that provides access by arginine 164 to the nucleotides so that the gap activity can be completed. The one on the left is our structural model. The one on the right is hypothetical, but we think that this is a close enough approximation to what the real complex looks like. But the question is, so what is this? What is the role of this complex? So so this is essentially a checkpoint, we think. It's poised to be activated, and when nutrient levels go up, this enables the completion of the gap activity. So what is this required for? So we are quite surprised that, in contrast to previous reports, knocking out folliculin in cells had no effect on the activity state of mTORC1. So here we are looking at mTORC1 activation by amino acids, and you can see normal activation in cells where we knock down luciferase, as well as if we knock down folliculin, as you can see here. So essentially, knocking out folliculin, replacing it with the wild type or the gap mutant had no effect, made no change in the activity of mTORC1 toward these canonical substrates, and also in the overall recruitment of mTORC1 to the lysosome. So where is folliculin important? It turns out that one substrate in particular, on the contrary, was absolutely dependent on the presence and activity of folliculin. And this is the TFEB transition factor that I mentioned before. So TFEB, just to remind you, is a booster of catabolism. It promotes lysosome biogenesis, autophagy, lysosomal degradation. And because it consumes cell mass, mTORC1 strongly and negatively regulates TFEB. So it phosphorylates it and prevents it from reaching the nucleus. So we then ask what will happen to the subcellular localization, particularly to the nuclear translocation of this transition factor where we knock out folliculin. And actually, the data I'm showing you here are with TFE3, which is a paralog of TFEB. So these are essentially identical or closely related transition factors that behave identically in terms of regulation by mTORC1, but we had a better antibody for TFE3. So what you see here is that in full amino acids, when mTORC1 is active, TFE3 or TFEB are out of the nucleus, so they're not found in the nucleus, and therefore they are inactive. If we knock down folliculin, we see that despite full mTOR activation toward the other substrates, TFE3 becomes completely nuclear. So it accumulates there, and we can demonstrate by qPCR that its downstream programs for lysosomal catabolism are activated. Moreover, this translocation is completely dependent on the gap activity of folliculin. So if we now reconstitute these folliculin null cells with a wild-type protein, we can restore the cytoplasmic distribution of TFE3, so we can pull it out of the nucleus, and this happens in an mTORC1-dependent manner. On the contrary, if we reconstitute these cells with a gap-dead R164A mutant, now you can see that folliculin is entirely nuclear, so it can no longer be pulled out of the lysosome. So the gap activity, again, is essential, and this is quantified here. So this leads us to a model that brings together all this data. So in minus amino acids or minus nutrients, the stable LFC is on the lysosome and poised for activation of the gap activity of folliculin FNIP toward RAG-C. This happens when RAG-A becomes GTP loaded, essentially breaking up the complex and enabling this complex to reorient itself and complete the activation cycle of the RAGs. Once RAG-C has been switched to the GDP state, we think that this is key for the recruitment of TFEB, TFE3 to mTORC1 so that the transition factors can be phosphorylated and essentially prevented from going to the nucleus. Loss of folliculin, for example, as occurs in Burke-Hogdubay syndrome, prevents this reaction and essentially causes TFEB, TFE3 to be constitutively nuclear. Could this be oncogenic? So could this constitutive nuclear localization of TFEB, TFE3 somehow promote tumor progression? So there is evidence that actually this could be the case in several cancer types. So in fact, the labs of Andrea Balabio, Lisa Hensky, and others have shown that the aberrant growth and aberrant signaling that occur in renal cell carcinoma and in Burke-Hogdubay upon loss of folliculin depend on TFEB, TFE3. So this nuclear translocation, this constitutive nuclear localization is key. Moreover, there is evidence that in pancreatic cancer, nuclear localization of TFEB, TFE3 can drive directly the growth of these cancers, and this could be related to the ability of these cancers to scavenge nutrients in nutrient-poor microenvironments due to the heightened activity of the lysosome. And so this essentially promotes the possibility that this switch or this follicle-independent regulation of TFEB, TFE3 could be a key step in the progression of at least some cancer types. More generally, a take-home message of this study is that there could be substrate adapters or certain regulatory factors, such as this complex here, that can tweak the activity of mTORC1 towards specific substrates. So far, we've considered mTORC1 as a general activator or regulator of all of these downstream programs without the ability to discriminate, but the folliculin FNIP2 and the complex they belong to could be the first example of a regulator that can turn on and off specific programs, achieving, for example, selective upregulation of catabolism, but in the context of intact anabolic programs. So finally, in the last few minutes, I'll tell you how we can use this detailed knowledge of how mTORC1 is regulated at the lysosome to come up with perhaps better or more effective strategies to modulate its activity either wholesale or towards certain substrates. So going back to the data I showed you before, I said how when we treated MPC1 knockout cells with different mTORC1 inhibitors, we saw different effects. In particular, rapamycin was not effective at restoring lysosomal function. And in fact, rapamycin, so the Easter Island-derived macrolide, the allosteric inhibitor of mTORC1, has had a lot of promise in terms of lifespan extension in several experimental models. Its derivatives, rapalogs, are used in certain human cancers. But it's not an ideal inhibitor because it's incomplete. So some of the substrates of mTORC1 are completely resistant to inhibition by rapamycin. In particular, rapamycin is not a great activator of autophagy. The newer ATP competitive inhibitors of mTORC1 are more complete than rapamycin. They can really block all of the outputs of mTORC1. However, they come at a cost of significant toxicity because they also inhibit a second complex, mTOR complex 2, which is essential for metabolic homeostasis, for example, for insulin signaling. And so the quest is still open for inhibitors of mTORC1 that can be both complete toward this complex, but also specific. And so to move toward the goal, we teamed up with Dan Nomura, who is a chemoproteomics expert at UC Berkeley. And so a postdoc in our lab, Regina, who I mentioned already at the start of the talk, and she's really amazing, teamed up with Claude Chong, who is a former postdoc in the Nomura lab. And together, they screened chemical libraries of cysteine reactive compounds for their ability to inhibit mTORC1 signaling. So as I mentioned, the Nomura lab has pioneered the use of cysteine reactive compounds that can block a reactive cysteine that might be found, for example, in the active site of an enzyme, or could be found at a protein-protein interacting surface, so we can use them to either block an enzyme or break a protein complex and so on. And so Regina and Clive use screen these libraries in the context of both in vitro screens, where we're looking at the assembly of recombinant complexes that are required for mTORC1 activation, for example, RAG regulator, or in cells based on induction of autophagy, which is a major readout of mTORC1 inhibition. And so they discovered two very interesting compounds. One is called EN6. This is a cysteine-reactive compound that targets specifically a conserved cysteine in the vacuolar ATPase. So this is the proton pump that acidifies the lysosome. And something I didn't mention before is that the VATPase is also essential for mTORC1 activation by nutrients. We don't fully understand how, but when this complex is dysfunctional, the mTORC1 activation process fails. And so this compound targets a specific cysteine in this complex. A second compound, which is still unpublished, CC144, targets a cysteine in the regulator complex, which is the complex that anchors the RAGs to the lysosomal surface. And so essentially, both classes of compounds, they target this lysosomal scaffolding complex that's required for nutrient sensing and mTORC1 activation. And as you will see, the mode of action is actually quite interesting and different from the allosteric or ATP competitive inhibitors out there. So I'll show you the data for EN6, but the other compound is similar, although there are also interesting differences. So here we are stimulating cells with amino acids, and we can see the usual activation of mTORC1 shown by phosphorylation of these canonical substrates. If we treat these cells with the EN6 inhibitor, we see complete failure of mTORC1 activation, and this is true for all substrates. Whereas importantly, unlike ATP competitive inhibitors, mTOR complex 2 signaling is not affected, as shown by intact phosphorylation, for instance, of serine 473 on AKT, a canonical mTOR2 readout. Moreover, treating cells with these covalent inhibitors causes a failure of mTORC1 to recruit to the lysosome. So up here, again, when we stimulate cells with nutrients, we see this beautiful clustering of mTORC1 on lysosomes. However, pretreatment with EN6 causes mTORC1 to remain diffused and not go to lysosomes despite the presence of amino acids. So this is a radically different mechanism from the other inhibitors, which have no effect on mTORC1 lysosomal recruitment. They actually affect the kinase function. This is a step upstream because mTORC1 can even go to the lysosome to become activated when these inhibitors are used. Also, quite nicely, these molecules work in mice. So here we took 3 groups of mice. We treated 4 of them with control, so with vehicle, 4 with rapamycin and 4 with the EN6 compound. And we look at inhibition of canonical mTORC1 substrate. So one substrate, S6, which is known to be rapamycin sensitive, is in fact inhibited nicely by both compounds. However, as you can see down here, 4-EBP and ALK1, which are rapamycin resistant substrates, so they're insensitive to rapamycin, are very effectively inhibited by EN6. Also, I mentioned that rapamycin is not a great activator of autophagy. So here we are looking at autophagy activation through lipidation of LC3B or degradation of P62. And so what you can see here is that EN6 does a much better job than rapamycin in activating autophagy. So this was true for skeletal muscle, heart, adipose tissue, and other tissues that we analyzed for this response. And so we were quite pleased in seeing that these new generation compounds can achieve effectiveness or efficacy and actually a broader spectrum of inhibition than rapamycin, but without essentially compromising other complexes such as mTOR complex 2. So we're excited about the possibility of further developing these new generation compounds, both for wholesale inhibition of mTOR1, as I just showed you, but also perhaps to modulate specific steps in the pathway. So for instance, the activation of the catabolic programs or modulating anabolic programs, as there could be a specific recipe that's ideal for either cancer or neurodegeneration or other disease settings in which mTOR1 is dysregulated. So with this, I would like to conclude and acknowledge the people who did the work. So I mentioned Regina, Oliver, and Chunyan, but really everyone in my lab gave important contribution to the studies. We have wonderful collaborators. And I'd like to acknowledge also our funding, and I'll be happy to take questions.
Thank you, Roberto. This was amazing and a lot of intriguing content.
Thank you.
Let me start off with one, and this is more of a conceptual, philosophical question, but I think what became evident by your talk, but I've seen this before, and it always strikes me how you have this very specific relationships where, for example, with FcLN1, you get effects on only one downstream target of the mTORC1 complex. And it's hard for me to really recapitulate that in the concept of this is activation of an enzymatic activity. So conceptually, how much do you think of what's happening around mTORC1 is really reorganizing of the lysosome as a signaling hub, and activities may not be necessarily purely driven by activating, inhibiting an enzymatic activity, and that also being why you have these disconnects between some of these inhibitors and the way you activate or inactivate mTORC1 is really important for downstream signaling effects.
Yeah, so I guess maybe one way to look at your question is to ask where and how each individual substrate is engaged in the cell. So first of all, the reason why rapamycin blocks certain substrates but not others is because the physical plug provided by FKBP12 seems to be sufficient to block some of them but not others. So it's the way they fit in the active site. There's a lot of machinery around mTORC1 in the lysosome, and so it could be that depending on how each substrate makes contact with this machinery, that determines under which circumstances it could be activated or not. We also don't know whether additional PTMs on specific substrates can make it a better substrate under certain conditions for mTORC1. And there is also another fact, which is, one thing that I didn't talk about is once mTORC1 is recruited to the lysosome, does it really stay there or can it move around the cell? And also, can substrate be recruited to mTORC1 with different efficiency, essentially? So all of these aspects, I think, are going to play a role. And it could be that depending on the particular cell type and disease conditions, these aspects change. So, we had this example of folliculin, but I think that that's specifically due to the fact that the RAG-C GTPase is being GDP loaded and then makes it an anchor for the T5 transition factor. Yeah, so it could be a lot more than that. Yeah.
Yeah, and I mean, it probably also is that you compete, substrates competing, and there's limiting activity of mTOR, and so how you guide things. But no, it's fascinating.
Yeah, that's an interesting idea too.
There's one question here from Raul. Roberto, impressive talk. Do we know how is the nuclear transfer of TFE modulated? Is it phosphorylated or modified otherwise?
Yeah, absolutely. So mTORC1 phosphorylates TFEB on at least 3 key sites. And so in that phosphor state, TFEB then binds to 1433, and that prevents it from going to the nucleus because it blocks its association with the importance. So that is the core mechanism. And so whenever mTORC1 is inactive, then TFEB becomes dephosphorylated. And there is actually work from the Balabio lab showing that the calcineurin phosphatase, which is actually regulated by the lysosome, has a key role in dephosphorylating the mTORC1 phosphorylated TFEB, so it can now go to the nucleus. There seems to be a lot more than that. First of all, there's other kinases that help to modulate phosphorylation on TFEB. There's other PTMs that have been proposed. So I think that that's just there's just a surface that we're starting to scratch.
Yeah. Another question. You've shown the NPC1 contacts that blocking activity of mTORC1 can be beneficial. If I recall your model right, it's really opposing between OSBP1 and NPC1. So would the proposal be that if you could selectively target OSBP1, you would even be more beneficial because you would not abrogate all of the other mTOR activity?
Yeah, so that's an interesting idea. We actually tried to do that, but both by knocking down, in fact, it has very strong inhibitory effect on mTORC1. And also there are natural products called orphylines that block OSBP. What we found so far is that these treatments are toxic. And I think the reason why they are, essentially the cells after a while die. And I think the reason why is because OSBP is also involved in ER to Golgi contacts. And actually, that was the initial role where this protein was described by Bruno Antoni and others. And that's probably important for secretory traffic and other activities that over time become compromised. So in order to block OSBP, so to block this pathway, we'll have to selectively decouple OSBP from the lysosome itself, as opposed to blocking it wholesale. And in fact, we are trying to understand if there is a co-receptor on the lysosome that anchors it there specifically.
Yeah. All right. I think we're perfectly at time. Thank you, Roberto.
Great.
That was fantastic.
Thank you very much for having me. That was a pleasure.
Thanks, everyone.